This is the second part of our discussion about how RNA, this heterogeneous RNA as we're calling it, is processed into mature RNA, mature message that can then be brought out into the cytoplasm and used. And we've talked about two major things that occur. One is a cap, a 5' cap, and then ultimately a 3' poly A tail is added. There are three parts of things that happen though. One is the cap, the other is the tail, and then there's what's called splicing. Splicing is by far the more complicated of these three processes and can occur co-transcriptionally as well, and it certainly can occur before the poly A tail is put on, or it can occur sometimes after the poly A tail is put on. But anyway, it's a major process and it's the most complicated part. So let's just think about what we have here. We've got a message being produced, okay? We get it with 30 or so bases put down. We get a cap put on, this G cap, modified methylated guanine cap that's put in. And as that process is going on, we can have splicing occur. Splicing is where you have removal of these structures we call introns. So splicing is simply the mechanism by which we remove introns and infuse together these exons. Okay? As it says up here, it can occur co-transcriptionally. There's a lot of evidence now that it does occur as soon as the message starts to be made and you get a length of that available, that the complex comes together and we start the process of actually having the splicing going on. But it also can occur, as I said, post-transcriptionally. So once message is cleaved off and the poly A tail is starting to be added, you can then simultaneously have splicing occurring. So it can occur at either place. But it's a very important process. Why? Because we want to remove these introns. Introns are these intervening sequences that we know, because remember, transcription itself is just a blind process. Once it initiates, it starts elongation, it just copies everything. It doesn't know which is an intron or an exon. It copies it all. So the introns are these intervening protein parts, or sequences that are not going to be involved in the protein parts. And then, of course, what we want to put out are these things we call exons. These are what are put out into the mature message, and these are ultimately what are going to be read and translated into the polypeptide that we're going to form later. Okay, and we're going to talk about the process up here, and we're going to primarily concentrate on what are known as spliceosomes. All right, but before we do that, let's talk a little bit more about these introns and exon ideas. Okay. It was about 1977 that some labs realized that there were these distinct regions within DNA. Okay. Back then we thought that DNA was, and that the genes were just genes, right? They were just a stretch of message, and that produced a message. You just read it all, and that was it. Well, it turned out not. That in fact, that there were split genes. By split, we meant that there were areas that were being read, but didn't ultimately make it out into the cytoplasm itself. And eventually a Nobel Prize was awarded for not only the presence of these units, but for the mechanism of splicing. And we've shown some pictures that look like this before, where you have complex, you've taken the message itself, RNA, and you've thrown it in and you've heated up DNA so it's single-stranded. You've thrown in a single-stranded message, and you look for where those hybridize, where they anneal together. And what you end up with are areas where you have both RNA and DNA and together, bound together, and then you have these places like the black here, this F or the D or whatever, where you have, they don't connect. This is single-stranded DNA. And so you have some large structures, it turns out, and you have some fairly small structures. And these bow outs, where you don't have a hybrid molecule of RNA and DNA, is actually where you have these introns. And you've seen this picture before when we talked about what a gene looked like and talked about introns and exons. So you have these forms. And what's important is that these loops, which are single-stranded DNA, some are very large and some are very small. Okay, so it indicates that for us that introns are not all the same length, right? They can be very different stretches. But what's important for us right now in splicing is that obviously this intron region up here, which was in the original message, and these are mature messages that are used for this hybridization, had to be cleaved out somewhere because they were certainly transcribed, but they never got actually put out. Okay, so what do we know about introns? Well, just what I said, the number of introns can vary between genes. It can be very few, and we'll look at a couple in a moment, or to very, very many. there's no set size, okay? And there's some signals that must be in there. So there's kind of a minimum amount of code that you can have because you need to signal the front end, the back end, and some other sites. But as far as the actual size and the number of nucleotides involved, it can vary greatly. So there really are no set sizes. And just to give you some examples of what I mean by this, let's go to mouse insulin. Okay, insulin. This is going to make a hormone, right? The protein eventually will be a hormone. And if we look at it, the intron size is in pink here, and the green represents the exons. There's only one intron here. It's about 119 bases long, and these are not base pairs because these are messages, right? And the total length of this message is somewhere around 562 bases. So 21% or so of this is of this particular message is actually a single intron, right? And that's not too big. It's very sized. And that's you notice this is a fairly small message. Is one of the reasons that insulin was one of the first of the messages that we could actually put into vectors and use in bacteria to produce the protein product that we wanted, which was insulin. And so one of the reasons we could produce it first was that, in human insulin being there. So for diabetics, we could make insulin that they could use. Up until that point, the insulin had to be extracted as a protein, usually from cows or something else, or pigs that were close enough to human that we could use it. So it was a big process of trying to isolate this product. But now we could actually take the gene itself, because it was very small, or excuse me, the message itself, because it was very small, insert that into a vector and end up with producing a process that would give us plenty of insulin, basically bucket loads of insulin, by following the process of inserting the DNA and then making message. Now, if we look at beta globin, we talked about that before. Beta globin, right, is part of the alpha and beta globin hemoglobin complex. And we talked about all the duplication and gene family stuff. Well, in it, at least in rabbits, there are two large introns. And notice that one of these introns is actually larger than any one of the other exons. So if we total that up, it all adds up to being more intron in this gene than there actually is exon. So in the message itself, you've got to cleave out more than half of the actual message and stick these three exons back together. And then just to take an extreme example, if we look at chicken egg ovalbumins, notice that we've got 1, 2, 3, 4, 5, 6, 7 introns. The introns are by far some of the largest pieces of this gene, of this message. And somehow we have to stick all these green areas back together. In this particular case, over 75% of what's actually transcribed has got to be removed from the system. And again, the two largest sections that we have are actually these introns that are going to be sliced out. Okay? So how do we do this? Well, we pointed out before that intron boundaries are highly conserved. And we talked about the fact that you have these special codes, consensus sequence, that tells us, okay, this should be approximately where we're going to have a splice, and this is going to be where the exon ends, and this is going to be where the next exon starts. So we have these very regular splice areas that we can use. There's also some important things. One's called a branch point. That will be important later. It's just an adenine residue that's found about 2/3 of the way down from the front part of the intron. Well, it depends on how long the intron is, but it's fairly close to the 3' splice unit. So this is going to be an area that's going to be important later on, because we're actually going to fold back part of this initial part once we cleave it, and this is going to be a place where it folds into. Right? All right. The next thing to tell you is that clearly introns are an evolved feature. Okay. Now there was a lot of argument early on about whether introns were there and then evolved out as we went to prokaryotes, or whether they actually evolved into eukaryotes. I think the consensus now is probably that they grew into eukaryotic systems. And one of the ways they know that is that if you look at places like viral DNA, okay, there's no introns there. Our mitochondrial DNA that we have in our system, as well as in plants, it's circular. It has no true introns in it, although there's some reasons, because it doesn't splice in that form. And CP, which is cytoplasmic DNA, which is found in in chloroplast DNA, found in chloroplast of plants, lacks introns. Now, interestingly, this has led to what's known as the Endosymbiotic Theory, is a sort of supporting for that. The question came about: we have the cell where did stuff like mitochondria come from? And clearly mitochondria is, particularly in eukaryotic systems, is very different. The DNA there is very different from the nuclear DNA, in that it doesn't have these introns and it functions very differently. But it turns out that there's been sharing between nuclear genes and mitochondrial genes. There's some mitochondrial genes that were originally mitochondrial, found in the nuclear regions, and there's nuclear genes that are found in mitochondria, so they're back and forth. But where did they come from to start with? Well, one idea is that the mitochondria, because of their shape and things, represent some bacterial invasions of early cells, and then they became symbiotic, right? Endosymbiote. Symbiotes means they rely. Mitochondria won't live by themselves outside the cell. Cells won't live without the mitochondria, which has now become the powerhouse of the cell. And then what about these green plants that can photosynthesize? Well, they of course rely on the fact that you have these chloroplasts. And chloroplasts have these very large circular DNA that's very different from the normal nuclear DNA of plants. And so the idea there is probably a blue-green algae that invaded these cells and made the cells functional as cells that could photosynthesize, and therefore gave the rise of plants. So that's endosymbiotic theory. The other thing about these introns is that they fit into what's known as exon shuffling theory. The idea is that if you're going to make a protein, and proteins work by the way they bend and function. So we'll talk later about the shape of a protein derives its function. You have left-handed bends and right-handed bends, and you have coils, and you have all sorts of things. Well, why re-evolve those over and over again? If you need a couple of right-hand bends, the code's already there. It's kind of like when we code, if you've ever coded programming and you make a subroutine to do something, well, you don't rewrite the whole subroutine. Next time you need to do a randomization of a number, you just grab that subroutine and stick it over here, right? Maybe make a few little changes to fit your program, but you get packages. And those of you that program in R language or something know that there are these little snippets that you grab and you use them for your programming, right? Well, the same thing here. Why not, if you evolve some systems, why not be able to shuffle it around? And so the idea is that the introns themselves serve as sort of safety zones or cut zones. So you can cut in those introns and move an exon around by moving the two introns on each side of it, or duplicate it or whatever, and you don't hurt the exon itself, the actual thing you need to code. So you can make copies, and you can be duplicated by occurring in the same gene. You can copy it and move it over to another gene and insert it there, whatever. And it turns out that within some introns now, we've learned during the last decade or so that there are these transposable areas. So they're clearly pre-primed for certain exons because the introns that surround them can cut and move and be transposed to make copies and be transposed over and to move those introns, those exons by transposition. So it's pretty clear that they are part of this. Evolution is being conserved there in the sense that you don't need to re-evolve it all the time. Move it around, make it different, and make a new protein by having this shuffling going on. Right? Okay. Now, how does all this work? Well, how is exon, whether it's shuffled or not, but how do we actually get this intron cut out? Well, there's multiple ways, but the primary way involves a structure we're going to call a spliceosome. All right, remember when we mentioned before that when you see some, whether we're talking about a nucleosome, now a spliceosome, and later on a ribosome, we're talking about proteins and either DNA or RNA complexed together. In this case, what we're going to talk about are a number of proteins coming in, and this RNA, which is the message that's going to be there, as well as some other RNA that's found in the nucleus itself that's going to help identify some of these areas that we need to splice. But that whole complex together is known as a spliceosome. And the spliceosomes, as I said up here, will form sometimes co-transcriptionally. They'll go ahead and form, or they may be involved post-transcriptionally as well, before after or during the time poly A tails are being put on after cleavage. So either place. They're large complexes, and they're quite complex, and we're still learning about them. Now, we definitely know they exist. We can physically see them, okay? And we can see them activated and around these messages. So this is a piece of messenger RNA, and this is a spliceosome that's conformed around it. So what are they doing? They're taking what we called heterogeneous RNA. Remember, we called it heterogeneous RNA when it first comes off of being through the transcriptional process, sometimes known as pre-mRNA. You may see it called that. And we're going to mature it into full messenger RNA. But what we mean by heterogeneous is it contains these introns that we want to get rid of, right? How this is going to happen is by a number of things. One are by some of these RNA molecules that I talked about they're called SNRPs, small nuclear RNA molecules, and they're associated with certain proteins, ribonuclear proteins that play this role, as we say. And classically, they've been identified as U's, U1, 2, 3, 4, 5, 6, 7, depending on which spliceosomes you're forming. Okay. Now, interestingly, just as a side note, there also turns out that some RNA molecules don't need all this spliceosome stuff to work. They actually can act as their own enzymes. And what will happen is the message will come along and it'll fold up and splice itself out and then recombine. So they function, essentially the message, the RNA, function as an enzyme. It was thought that only proteins could have enzymatic activity, but it turns out that RNA can actually have enzymatic activity as well. And that was worth a Nobel Prize in about, I think it was about 1989 or so, because, no one thought that a piece of RNA could somehow have a catalytic function. And it fit right in with this whole egg, chicken or egg argument. What were the first biomolecules of life? Did we start out as a primordial soup with making proto amino acids, and those amino acids string together? People say, oh, but amino acids don't really carry information. So how did that work as far as replication and things? And other people say, well, DNA probably wasn't first because it's fairly complex. Maybe just single-stranded RNA was the first biomolecules of life because it could be replicated, it could form, and and they say, well, it can't have any enzymatic activity. It just sit there forever. Well, maybe it did have enzymatic activity. Anyway, it's an interesting argument about where proto-life comes from. But our point here is that there are some times when RNA itself can act as its own splicing elements. Okay? But for right now, we're talking about taking heterogeneous pre-RNA, messenger RNA, using SNRPs and using a bunch of other enzymes for it to work. So here we've ' our message, right? We've got an exon where we've got an exon recognition site on the 5' end, and then this exon 2 site on the 3' end of the intron, and we've got our branch point. Okay, we're going to start bringing in these elements. Okay, the first element that's going to come in is U1, and it binds up to this 5' site up here. Then we bring in U2, which will bind up actually to the branch point. Okay, and then we have a whole bunch of elements that come in. 4, 5, and 6 actually come in. They're all recruited once U1 comes in. Okay, and they grab a hold, and what they're going to do is help cause this formation of a loop. At that time, U1, U4 are usually released. We don't have to get too worried about exactly what's happening here. Our point is we have a number of these ribonucleotides that are coming in, and they're bringing in enzymatic activity, and they're starting to help form a loop. And that loop is formed as part of this branch point that's coming along here, okay? So we disassociate these two, leaving this sort of heterogeneous RNA. And then finally, they have an affinity for the U2 and the U6, and they will go through a transdispersation process, which basically causes them to cross bond or pull together and form up a bond. Okay. All right. So once we have that forming, we end up with what's called a lariat structure being formed. Okay. Lariat structure is a loop. So, a lariat is the old cowboys' rope that they would use to rope cattle or whatever. It was called a lariat. So this looks like the ends of a lariat loop right here. Okay, so we start to have that formed, and once we do that, we cleave this first site. All right, so we cleave off this end of the exon intron boundary. The 5' end of the intron is cleaved off, and we have our loop then move. Now there's a lot of enzymatic activity going on here. Okay, it's not just all of it. Okay, we go ahead and we actually then cleave this side, because what we're going to do is pull down this 5' end. We're going to be going through ATP hydrolysis. It doesn't really matter, but we're going to both cleave that off. And because this is a large complex, it's going to be large enough that we're going to be able to bring this exon end and this other exon in together, where you're going to have a fusion. You're going to have a reconnection. So you've got this, we're basically just talking about single-stranded RNA here. So you're talking about the fact that you've got these phosphodiester bonds that you have to reconnect, and that's exactly what happens. Okay, we have the formation of this, right? And then ultimately the SNRPs are going to be released, and you've formed this together. You've brought the two exons together. You've reformed a phosphodiester bond there, and it's just single-stranded RNA. These pieces go off, they release. This is not protected anymore, right? There's no cap, there's no poly A tail, and so these nucleotides can be broken down. They can be rephosphorylated and recycled for the next time as we move through. All right. Now it's a complicated process, as I said. This is kind of one big picture of it. Here's our original piece of message, a heterogeneous message. Here's the end we want to start with over the exon 1. Here's exon 2. Here's talking about bringing in the U1s. We start to fold it back, and it shows you how you kind of get this fold into this lariat structure. The spliceosome then forms this large complex by bringing in all these other elements. You then start to have cleavage and formation of the lariat. At that point, the first exon is close and is attracted to the second exon. There's events that goes on using ATP. You get resplicing going on. The lariat's released, it's broken down, and then the splice exon here. And then all these units are available to cycle back down the line to the next exon intron boundary so that they can remove the next introns. And this goes on continually all the way down the message until you've removed all the identified introns. Okay? And that's the formation of a spliceosome. A spliceosome is this large functional unit right here that we see made up of all these elements. And these elements, once again, are they're nuclear RNAs, so they're small nuclear RNAs, SNRPs, that help identify these points. They sort of bond up across these. And they are also obviously enzymes that come in and have to come into play that help do some of the cleavage, breaking apart, as well as the reuniting that forms up here. All of that is spliceosomes. That's one of the primary things that's happening. It's constantly going on. It's happening in your body right this second, going over and over and over again, because we're constantly producing proteins, and to produce those proteins, we have to have message, and we have to have that message matured so they can get out and function, right? So now we've gone through all three things. We've capped one end, right? We have gone through the process of removing these introns and pulling these exons together, and we have added a poly A tail. So now we've gone from truly heterogeneous message to mature message that's ready and signaled to be transported out into the cytoplasm. Okay? All right, that's where we are. Now, in the next lecture, though, I want to talk about, yeah, this works, but there's some nuances to all this. I said early on when we talked about introns and exons that sometimes an intron can be an exon. Sometimes exons are not all used, right? They can be skipped sometimes. And that all comes back to the fact that we've got maybe 20,000 functional genes, these things we're calling classic or traditional genes in our system that are eventually going to make proteins, but we've got at least 100,000 proteins. So somehow this splicing has to be involved with picking and choosing and making up these different messages that are going to work, because we don't have 100,000 places we take message from in our DNA. We have to take the messages that are coming off of these 20,000 and convert them some way to give us all these different proteins that we need. So that's what we're going to talk about in our next section.